Today we are going to be looking at the biological practical exam. We're going to look at generally what we are likely to be expecting in this very exam, and I want you to pay attention and watch to the end, because there are some of the sensitive experiments that will show here. I will display it here live so that you'll be able to see them and be sure that actually this is possible based on what we have here. So here in the specimen, the first statement we have the specimen A is a groundnut pod. Actually it's groundnut pod with seed. I was just trying to make more space, create more space. So that groundnut pod containing seeds. Specimen B is what? Crushed or milled roasted groundnut seed. That's what specimen B is. And then specimen C is a beaker with 20 ml of water. Then specimen D is beaker with 20 ml of table salt solution. Then specimen E is a beaker filled with sandy soil. Specimen F, a portion of alimentary canal of a small animal from esophagus to the end of the stomach, meaning that it's like we're going to stop at the stomach and leave the rest. End of the stomach. Once it ends at the stomach, the rest of them might be, you can leave it behind. So the next one is specimen G, which is a twig or branch of plants with Mistletoe. The next one is specimen AH, which is evaporated milk. Then specimen J is fresh fish. Specimen K is a dry maize grain. Specimen L is tomato paste. Specimen M is tomato fruit. And then specimen N is rotten tomato fruits in petri dish, and the tomato paste will also be in petri dish. And then we look at the specimen P, which is a thermometer, which we have here. Specimen Q is a meter rule, and finally specimen R, which is what? Quadrat. So quickly we're going to be analyzing some of these things based on what we are likely to expect in each of them. In the specimen A, let's start with that. In specimen A, we are given what? Groundnut pod with seeds. Now, one thing I want you to note about the groundnut pod is what? A legume. Is a leguminous plant. Now, they play a very important role in the soil because they have a ability of partnering with some bacteria to fix nitrogen in the soil. But that's one of the important things you should note about them, because you never can tell where the question will be coming from. Now, another thing you should also look at is the kind of germination that occurs there. The type of germination is the germination we call epigel germination. This type of germination, the cotyledon moves up from the surface, comes to the surface with the plant that is growing. And also this is example of what dicotyledon. That means they have two seed leaves. You should note it. Dicotyledonous plant. So they also have the type of leaf they have is a net venation. That is what they have here. Net venation. We have a net venation. That is their net. And then another thing you should note, the type of rooting system is what? Taproot system. Now we can also use it to compare coming down to this place. It's likely that they may ask you to compare with this, where we have the monocot, which is a kind of different from what we have there. The rooting system of this one is what we call the fibrous rooting system, while this other one is what? Taproot system. In fibrous rooting system, they don't have any given roots. The fibrous look like this. Now I'm comparing two of them. Then the other one is taproot system. In taproot system, they have literal roots and then one solid one that is going downward. Now, in their vein, in this maize, in the maize seeds, the kind of vein they have is parallel, where all of them, the veins are like this. You understand? And it's common in what? Grasses. That while this other one is more in what we have in dicotyledon plants. So which other thing are we going to look when we're going to compare these two? Whatever I said, okay, this other one contains protein, while this other one contains what? Starch or carbohydrates. This is another good comparison between them. So now we move on to the next specimen. The next specimen we have the crushed roasted, I think yes, called crushed milled roasted groundnut seeds. Now here, since it's crushed, and what I'm thinking that will come out here is a situation where you'll be asked to test for what? For fats and oil. Now by having that in, so in the exam you're expected to have a filter paper. The filter paper now can also be used in testing for what? The fat and oil. How would we do that? You get the grain and put in filter paper. This is my filter paper. I've already added something there. So when you add it in filter paper, you can allow it to stay for some time. I just need to press it a bit so that whatever the oil can come out fast. I have a crushed one there because if you allow it to stay, actually this is exactly what will happen. But because of time, I will just want to see how far we can get. So now I was talking about how to test for this oil. Now what happens is that at the end of the experiment, with the filter paper, this is what you will observe. I don't know if it really appeared in the other one side of it. I never knew that the thing was off along the line. Why is off? I don't know. So look at this is what it looks like. See the place where the oil is, where the groundnut is a kind of translucent, allowing light to pass through more than the other side. So you can see how it's like from here. This other side, no oil. This place now, the center here is where the oil is. So this is one of the ways of testing for fat and oil in something. Because now the next one we're going to be looking at is what we can do with this beaker of water that we have there. You know what water can do? Water is very important in the transportation of nutrients, this digested food and mineral salt in plants. So this is what it does. But then another thing I think we can also look at, besides the importance of water, then the next thing we should be looking at is what we are going to do here in these pests. Tomato pests, we're going to be testing, and when we get here, we're going to test for it, show you how it works. I'm going to show you how it works. So my mic just dropped. So I'm going to show you how it works when we get to this point. The mic is no more working. I believe that I'm loud enough. When we get to this point, the pest will need this water, this specimen C, when we get to this point. And then I think we will also need it when we get to the hedge altogether, when we're looking at the evaporated milk. So let's quickly go ahead and continue with what you are doing. Now we'll go over to the beaker containing, beaker with salt solution and table salt solution. Then in that case, what we have here, we'll be using it to study, that salt always have to maintain osmotic pressure in a environment. It creates a hypertonic environment. It's an environment of highly concentrated solution, thereby causing osmosis to move from water to move from the lower hypotonic to the region of what? Hypertonic solution. So I believe that we are going to be doing this here. I think I will do it here now, leaving it until I have a rotten tomato, because I believe we might be using this when it gets to the rotten tomato. I have a rotten tomato. I will put my salt solution. This is my salt solution. I will put some quantity of it here, so that when the time comes, when it gets to that point, we will then check what will happen in terms of water moving from the other end. What I believe that will happen here is that the tomato will shrink more. The rotten tomato will shrink more because um, the the tissue there will serve as what semi-permeable membrane, allowing fluid to move from inside to outside to meet the concentrated solution of what hypertonic environment of this salt. So let's continue, and then we'll come to the sandy soil. The sandy soil, what I believe here we'll face here will be, what we're going to face here will then be looking at the texture, structure of this place, of the soil. In sandy soil is always has low water retention, that is water holding capacity, low water holding capacity. Another thing is that it's what coarse, or you can say it's what rough. It's also less, it has less nutrients, low nutrients content. Another one here is that, what's the other thing am I remembering? I can't remember any other thing. So these are the, I think these are the few words I can remember at the moment. So that's as much I can remember at this moment, please. Let me not crack my brain so much. So we'll go back to the portion of elementary canal. This canal, what we're doing here, what would be should be expecting here should be, what we should be expecting in this, he said that, from what I have here, he said that elementary canal from esophagus to the end of the stomach. So we're going to stop at the stomach only. If it is esophagus, no, esophagus, this is the stomach that look like this. End of the stomach is somewhere around here. So what it means, we are dealing with whatever we have here. If we are dealing with that, you know that esophagus is a channel that connects the mouth and the stomach. Now what happened in the stomach, we have what we have digestion of protein starts there, and inside the stomach we have the enzyme we call renin and the pepsin that add on protein. This add on protein that we get from milk, and this other one is any other protein. Pepsin, renin is for protein from milk, pepsin is for any other protein, and it has acidic medium here that supports their activities. So this acidic medium creates environment that activates the activities of these enzymes. So these are the things I think we should be expecting in this area. So the next one we're going to be looking at is the specimen G, where we have a branch of plants with Mistletoe. Mistletoe is parasitic plants. It's a parasitic plant that always attach itself to other plants. It has what we call a sucker. This sucker helps it to derive nutrients from the host. So one of the things is that at the core, when they are so much like that, when the process continues, they tend to destroy, kill the host. So this is another thing we should also be looking out for. So the next one we're going to be talking about the evaporated milk. Like I said, here this is where the specimen C will play one of the roles, I believe, in this experiment, in this practical exam. In this place, we have specimen H is what evaporated milk, and we have it here. So what are we going to do? We have, from the requirements, we have sodium hydroxide and copper sulfate, which must be required. You need to have it on your table when you're doing this kind of this test. So I have my milk here. So it simply means we're going to be testing for what? Protein in milk. So one of the things you use is copper sulfate solution, which they say we should be made available. I add a copper sulfate solution into this milk. We also have sodium hydroxide there. Sodium hydroxide that will be made available to you in the exam or on the exam day. So we add sodium hydroxide too. So you see the purple color that appears. This confirms the presence of what? Protein. That is one thing you should know about. This confirms the presence of specimen C plus copper sulfate plus this, adding to this plus copper sulfate solution and sodium hydroxide give you purple color, meaning that protein is present in this milk. So that's the thing. So I said again, this specimen C should also be playing a role in this when we get to this point. I want to encourage you to, stay with us while we get there, and we should also do that. So we're going to the fish. In this issue of fish, we are going to be looking at some of the features. I think since we are done with this, I can comfortably clean this area so that we can make use of it. So the fish now, I believe that we might be drawing the fish. So in most cases these days, they will give you the length you want. Sometimes you have 8 to 10 cm or 6 to 8 cm. Whatever may be the case, make sure that you don't go beyond this number. It must be above. It simply means that it should be more than 8 cm, equal to or more than how do I put it again? This should be, you see that equal to 8 cm or more than it. This other should be 6 cm or more than it. So you measure out what you want with your ruler, or you can make a square, 8 cm square, and put your drawing inside. Let's say this 10 cm. So whatever you draw from here to here will give you what you want. But let me just make a sketch and use it to show you some things that you might be expecting. Assuming this is my 10 cm, and then I want to make a sketch of my fish. This is my fish now. My fish should not exceed this place. As you can know, it should not be more than this side too. So what am I doing here now? So this is my fish. This drawing should be a freehand drawing, free sketch drawing, free hands sketching, and it's advisable to do it with what? 2B pencil. 2B pencil is advisable to do with 2B pencil. Now look at what we have. So here we have the gather fin. We have the anal fin. We have the this should be the pelvic fin. This is the what? Pectoral fin. This is the scale, and this is the dorsal fin. This is the lateral line. This is the ocular column. This is the eyes, and then basically, I think we're done with this one still. That the eyes, and then we have the mouth too. So this is just a sketch of what you are expected to know. So we have in fish, this the lateral line is what helps it to be sensitive to the environment, helps it to grab the activities any action that's going on in the environment. The column is where what we have inside it is a cover, the gray cover, the gill cover. That's what we normally know it to be. Now we have the pectoral, we have the pectoral fin, we have the pelvic fin, we have anal fin, caudal fin, and dorsal fin. Now we have the mouth. We also have the nostril. Nostril. So we also have the nostril there. So these are some of the things I think we should be conversant with. So in fish, it has three unpaired fins. Three unpaired fins. Three of them is the one we call the dorsal. The dorsal fin is not paired, it's single. And we have the caudal. It's also single. We have also anal. It's also single. Notice they are unpaired. Now they have two paired fins. The one of them that is paired is the pectoral fin, and the other one is the one we call the pelvic fins. These are the ones that are paired. They are paired. There are two here and there, but this one is single. So the function of this dorsal fin is to maintain balance when it's moving in a high speed. This caudal fin is to propel forward. And this open and closing of the ocular column helps to, for respiration. The eyes is for sight. We have the mouth which is for feeding, and the scale which has a kind of a protection to it. So you can leave it here for now. And the features that help it to survive. These are some of the things, the function of the features, adaptive features. There's another that I want you to note here. They have streamlined body, which help them to move very fast in their environment, in their habitat. They have streamlined body. You should note that too. So we'll go back to the dry maize. The dry maize now, I've already compared it the right side. So in this place, we can also look at testing for starch, because iodine will also be provided. This iodine, crushing this maize and then drop iodine on it, will turn it to blue-black. Then confirming the presence of starch. Then for the tomato paste, we'll go over to that place. What you'll be expecting there, because we need Benedict solution there, and then we need to be testing for we should be testing for sugar there. So that is with the paste. We put the paste inside the test tube. Then we add some quantity of water. So then we add Benedict solution. Let me reduce the quantity of this. I need to reduce the quantity. So I add Benedict solution. Then it will need to be, it needs heating. I'm going to be heating it now. let me hit it. I'm hitting it now. The color has changed. That is telling you that what we have here is what? Sugar, reducing sugar. That's monosaccharide. Has changed to orange coloration. It's evident that we have monosaccharide, that is reducing sugar. So the next one we'll go over to the fresh tomato. In the fresh tomato, the fruits, what we should be expecting there, we have to be expecting one, the type of fruit we have there is what we call berry, and then the next one you should also notice that is what in presentation of tomato is what acid presentation, and the acid is the type that's like this, where you have all the seeds. Most of the seeds, some of the seeds are in different chambers. This is the seed. They are in chambers, like the one you have in a, just like what you have in a, similar to what you have in orange. So in this chamber, we have each of them tilting towards the center. We have this. This is the epicap. We have the mesocap, then endocap, and then finally we have the seeds there. We have the seed. So this is a kind of transverse section of tomato fruit. So this is what is like, and that's what I think you should be looking out for. So the rotten fruits, we have it here. Actually, the thing has shrunk to some extent. It has shrunk. It's coming down somehow, and the fluid inside is also coming out, making this water to change to some extent. It's changing the color of this water because the osmotic pressure outside is stronger than the one inside now, making the water to come out of this very tomato. And see how strict the thing is at this moment. So this is one of the things that we can also expect there. Another thing we can also look at how we can avoid this rotting. We can avoid it by converting it to paste, or canning it. You can can it to preserve it for a longer time so that it will not rotting or spoil. So the next one we have is thermometer. Thermometer is what we use in a study of ecology. In fact, from there to the last one is what we use in a study of ecological studies. That is what we do with all those things. So here, what thermometer is used to measure to check the temperature of the habitat, the soil, the water, as the case may be. That's what we use it for. So the meter can also be used in checking calculating the area of a habitat. So the last one we have there is a quadrant. Quadrant is used to study the population of the environment. Perhaps also we can also use it to study population density. So now I want to let me just give you an idea of how to use this quadrant in solving problem that has to do with population. For example, we toast the quadrants, and each of the quadrants, wherever we toast, we check, calculate the number of organisms that we're able to see. Will be plants, mainly plants. So for example, after the toasting, we had about 20 toasts. Number of toasts is equal to 20. Then total number of organisms in this toast, number of organism is what we can say. Let's say we have total of maybe 500. That is what we have. Now we have the quadrants. The quadrants we use here, I can say that the area of the quadrants is what? 1 meter square. 1 meter square. Now to calculate, we can use it to calculate the population density. Pop density. Pop density is what? Number of organisms. Okay, before we do that, we need to find the area average. We need to use the average. But then let me go ahead and do for number of organism over what? Organism over what? Area of what? Habitats. Now let's get the average. Getting the average of this, average is what we need. We have 500 over what? 20. That will give us 25. 25. So to calculate the population density, number of organisms, which is 25 now, all over area of habitat, which we use our estimated area, which is 1 meter square. That will give us what? 25 organisms per meter square. Now we have our population density. For example, the real area of the organism we are studying is, let's say, is 100 meter square. Area of habitat, the habitat of the organism we are looking at, let's say, is equal to what? 100 meter square. So what it means now, we can substitute it here. That will give us number of organisms. Now we don't know the number of organisms in this very area, but we now have estimated population density, which is what? 25. 25 is equal to number of organism over what? Area, which is 100. So when we cross multiply this, we have the number of organisms, our estimated number of organisms here, and they will then be equal to 25 times 100. In fact, so that will give us about 2,500 organisms. That's how we use this. So we'll be able to have an estimate of the population of the organisms by what we have done so far. If you're able to get to this point, thank you so much for being part of the class. And if you have any question in regards to this, you can leave it in the comment section. I will always attend to you. And I encourage you to like our video, share, so that you can get to more people. If you are joining us for the first time, we encourage you to subscribe, and then see you in our next video. Thank you.